So yeah, today I'd like to talk to you specifically about metabolomics, what it is, and how we can utilize that with other multi-omics to understand disease, and particularly with the underlying idea of epidemiological study design. But to give us a little bit of context, I want us to go back in time. The year is 2003, and I want us to just think about how much has happened when it comes to multi-omics in the last 25 years or so. In 2003, we had Eric Lander and others. We had the Human Genome Project, and it took over 10 years and $3 billion to sequence just one human genome. And I remember sitting there in April listening to him lecture, and this was certainly a key point in time in this field. It wasn't that much later that James Watson, who was the one who discovered the double helix, was able to get his entire genome sequenced for just $2 million. At that time, in 2007, the vision of where we were with genetics specifically can be best summarized in this phrase. That was in Technology Review in 2007. Scientists ultimately hope to bring down the cost of whole genome sequencing to less than $10,000, a target price that many people believe will be the turning point in genomic medicine. At that price, many could afford to have their genome sequenced, and doctors could use that data to give their patients more personalized medical advice. And this really encapsulated the idea of precision medicine and what it was in the year 2007, with really just the idea of genetics behind it. I want us to fast forward until today, when we can get our entire human genome sequenced with a little spit of saliva or a drop of blood, and we're now in the range of doing this for hundreds or low thousands of dollars for just one human genome. Along with that, what has developed when we think of genetics throughout that period of time, a lot of different individuals and researchers went to study human disease and identify genetic variants associated with human disease. And I like to say that throughout that 20 years, we developed what I call the omics standard. And that is, what do we use to actually identify and validate and substantiate a genetic effect? And through these genetic studies, we identified that we need large sample size with replication in independent cohorts that is robust to multiple comparison correction. And then we actually need to have further biological interrogation of our finding to actually see if the association with the specific genetic variant is causal or not, and where the causal variant is. So indeed now, when we look at where we are, and when we look at what's happened in this past 20 years, we are no longer thinking just in the context of genetics, but we're thinking in the context of the whole central dogma, that is, whether it be DNA, RNA, protein, or small molecules. And that is really where metabolomics now fits. Metabolomics is the study of small molecules in a specific biological fluid. And I really want to talk to you today about how we can utilize metabolomics, sometimes by itself and sometimes in conjunction with other omics, to really translate what we see with omic variants to a precision medicine framework. Now, why do we think about this with metabolomics, and how can that help us with underlying other omics? I think it's important for us to realize that the omics happen through a cascade of events that we learned about, early in our biology career, where we know DNA goes to RNA, goes to protein, goes to small molecules. And while we initially focused very much so on genetics, there are a few things I think that are important to realize as we go from the genome to the metabolome. And one is, while the genome is fixed, as we go down this cascade, we slowly enable environmental influence to be a part of the equation. So it is now not just our genome, but it is the combination of our genome and the environment together that is dictating what either our proteome or metabolome may look like. So when we think of precision medicine in 2020 versus what we thought about in 2007, we really think of this in a distinctly different way. Rather than just being genetics, we now have a much broader framework where we look at multiple omic variants in combination with clinical data and lifestyle/environment to generate more precise precision disease phenotypes. And what exactly do I mean by that? Well, initially, if you look on the slide all the way to the left, we would have a bunch of people who were diagnosed with a disease, let's say hypertension. And of those people, we would give them medicine. And some of those people would have a great effect from the medicine. Some may not have that great of an effect, and some may actually have a bad effect. We transitioned from that to what we call stratified medicine, where basically we created different subtypes, usually based on clinical data or underlying risk profiles, maybe one biomarker, but really not a lot. And then, utilized treatment options that could be specific to those generalized class of subgroups. In precision medicine, we look at a much more comprehensive framework. That is, we look at all of some of the underlying omics in combination with all these clinical data and potentially biomarkers to then inform the optimal treatment for each individual. Now, when I talk about some of the studies and what we've done with metabolomics and other omics, I also want to point and emphasize the importance of study design and how that can impact the inferences that we make from any given study. Especially the concept of large population-based studies and the idea that we can utilize multiple studies together to inform precision medicine. Primarily because that capitalizes on many things, from study design to multiple visits to a larger number of biological specimens and omic data and multiple cohorts that enables us to assess things like generalization. So in what I'm talking to you about today, I just want to give you an idea of what we have assessed and grouped together, even for what I'm showing you today, for specifically metabolomic data. And when we look at this, when we utilize multiple studies, multiple study types, multiple studies, multiple individuals, multiple time points, multiple types of biospecimens, the total number of samples that we're looking at is much larger, right? We go from any individual study that may only have 750 samples to 25,000 samples. So just by doing this, where we group cohorts together, we have the ability to much more robustly assess different associations. And in fact, when we look at these different types of study designs enable us to infer different things. So the combination of multiple study designs together may enable us to look at things, and I will be speaking about asthma today, that may start from underlying asthma etiology to looking at the pharmacometabolomic impact, and finally to be able to translate that into clinical practice. So with that, I'm going to go over two general groups of studies that we've looked at that talk about different aspects of this. The first part of this is going to talk about a few cohorts and the idea of using longitudinal multi-omic data. Now, while the focus of this is on metabolomics, we also in this study and in my examples, I will illustrate how we've incorporated other omics, whether it be genetics, microbiomics, or outside exposures. The second part is going to really take utilizing that and looking at it in a framework where we can actually take metabolomics all the way from the discovery population through to clinical trials and finally to biobanks and precision medicine recommendations. So in starting with the first part of this study, where I'm going to be talking about really utilizing the multi-omic data, I'm going to be talking about a study called the Vitamin D Antenatal Asthma Reduction Trial. And this was a study that was born by my mentor, Scott Weiss. And I actually remember this vividly. We were we were at a wedding, a colleague's wedding, and he looked at me and he said, "Jessica, I really think that giving women vitamin D during pregnancy can reduce the risk of asthma in their offspring. And I want to do a study on this. And when we do it, I want to look at all sorts of things, multi-omics, environmental exposures, etc." Little did I know that that conversation would then progress to now what is 13 years of really exciting research. And why did he think this? Well, just to give a little bit of background, one of the things that's important to understand is vitamin D plays this critical role in the immune response that may reduce inflammation, which develops during asthma. And particularly the prenatal period was a time that seemed to be very susceptible to this, as that's when lungs grow. And so the influence during that time and making sure the supplementation was correct, he believed was key. So I'm going to give you a little overview of this study. This study started with women early in pregnancy, and they were randomized to either the standard dose of vitamin D or the standard dose plus 4,000 IU. And we've now followed those individuals. Those kids are now around 10 years old, and we've looked at the development of asthma over that time. Now, just to give you a summary, this is the initial publication of this study, and we actually had a sister cohort who did something very similar in Denmark. And when we looked at these studies, sure enough, what we do see is that there's a protective risk of vitamin D that reduces asthma by approximately 23%. And while in each one of these studies, they were quite different in several regards, especially ethnic makeup, socioeconomic class, this reduction in risk was profoundly similar. So what else did we do in this study? Well, one of the things we did was we generated a lot of multi-omic data throughout the course of pregnancy and the children. And I'm going to be focusing specifically on metabolomics. So I want to give you an idea of what I mean when I say we have generated comprehensive metabolomic data. These indicators down here show you the time points that we have generated either plasma metabolomics. You can see they've been generated in the mothers and in the children throughout the course of their life. The other thing we did was we partnered with our COPSAC friends in Denmark and actually looked at both of our studies together and asked the question, can we actually standardize time points that we generate these multi-omic data so that we can actually look at things together and replicate and validate results, which really is a key, I think, when it comes to omics and validating our initial findings. So I now want to talk to you about metabolomics and multi-omics. And when I say that, the first multi-omic I'm going to look at is multiple metabolomes. And that idea is this question: can we look at multiple metabolomes in the mother during pregnancy and understand anything about asthma risk in their children? So we had 2 time points, early in pregnancy and late in pregnancy, when we generated metabolomic data on the mothers. And we assessed asthma at age 3. Now, these were done at age 3 because at that time, the kids were younger. So that's why the age is a little bit younger there. So I'm now going to show you what we saw. So the results for the metabolome, both in early and late pregnancy, we saw a bunch of different associations. Now, what you see here is this is the odds ratio of 1. Anything above is an increased risk. Anything below is a protective risk. And one thing I do want to point out is during pregnancy, we understand that embryogenesis has different influences at different times. So it isn't necessarily unusual to see different influences early in pregnancy and late in pregnancy. So for example, what we see is later in pregnancy, we see a protective effect for both cortisol and cortisone and some sphingolipids, which didn't surprise us because we know later in pregnancy is when the lungs develop. And we saw this increase in sphingomyelins, which are also related to these sphingolipids. We were able to replicate that in COPSAC. As I said, we try to replicate results. But the other thing we found that was quite interesting and seemed to have a consistent negative effect or protective effect on asthma was this one group of metabolites. And this particularly struck me because one of these metabolites was called theophylline. Now, for people who are in the field of asthma research, we know theophylline. We've heard of theophylline. And why is that? Well, for me, this struck a particular chord because I have asthma. And if I think back to my own life, back in the summer of 1984, this is a picture of me in the hospital where I was hospitalized one of several times for severe asthma. And what medicine was I taking at that time? Well, I was actually taking theophylline for asthma. Unfortunately, theophylline wasn't really cutting it, and I was in the hospital, as you see here, in an oxygen tent. Having said that, though, when I saw theophylline, and I saw that increased amounts of theophylline in these pregnant women were associated with reductions in risk of asthma, I was particularly intrigued. So, as I said, what do we know about theophylline? As something that has been known to treat asthma, it is a known bronchodilator. It increases the size of the bronchioles. It also has anti-inflammatory effects. But the other thing that was particularly intriguing about this is theophylline is not commonly used today. When you see that picture of me from 1984, it was used all the time then. But now this is really not something that is commonly used. So why are we seeing higher amounts of theophylline in pregnant women associated with reductions in risk of asthma? Well, when we looked at this further, we found something very interesting. And that is theophylline is a primary derivative of caffeine. So pregnant mothers, looking through this further, the question is, are they increasing their caffeine intake? Are they drinking coffee and having chocolate? And is that actually increasing these levels of theophylline? And then the other question was, why didn't we see caffeine? If we see theophylline in the metabolites, why aren't we seeing caffeine? So we identified all of the particular caffeine metabolites that we had in our data. And sure enough, we see them here. There's in various aspects from what we see in the liver to the gastrointestinal tract to what's in actually ground coffee. And sure enough, we see in this that several of our metabolites highlighted in red here all had a reduction in risk of asthma. Actually, it was these three, apologies. But then the question is, why was caffeine missing? Well, caffeine was actually missing because in our QC step, we took out metabolites that had a lot of missing data. And when you look at pregnant mothers, one of the recommendations is to not drink a lot of coffee. So this was missing in many of the pregnant mothers, and it was actually dropped from the analysis. However, when we looked at it again, when we brought it back in, we did see, again, this protection of risk of asthma of caffeine as well. In looking at this further, we actually see that this protection against asthma for coffee, it has been observed before. So in this study, I just want to highlight that looking at the multiple metabolomes, we can see how things change over time. And we can also then look at our biological hypothesis in the context of changing time. We also replicated these results in other cohorts, which fits in with our concept of generating robust associations. I'm now going to just very briefly give you an example of a few other multi-omics studies that we have done. Unfortunately, I can't go into the details because of time, but just to give you an idea of the other types of things we can look at. And in this study, one of my colleagues and mentees, Kathy Lee Sarwar, actually looked at multiple metabolomes. She looked at the plasma metabolome and the stool metabolome in conjunction with a microbiome, dietary intake, and looked at all of these together for risk of asthma. Using co-inertia analysis, what she discovered was that when we look at any of these things, food, the microbiota, diet, and plasma metabolites, a large portion of their effect was being mediated through the intestinal metabolome to cause asthma. And the mediation percentage was about 25%. And that, in fact, things that were highly influenced in this pathway was dietary intake, the intestinal microbiota impacting the intestinal metabolome. Things like reduction of lack of breastfeeding associated with increased asthma, also impacting intestinal metabolites to cause asthma. So highlighting this interplay between other omics on one side, the intestinal metabolome as a mediator prior to disease. Another example of multi-omic data that followed very logically from this study was this question. Can we look at genetics in conjunction with the metabolome in the context of this VDART study where we also had a known exposure of vitamin D and look at all three things with risk for asthma? And why was this particularly compelling? Well, it was particularly compelling because one of the underlying, the strongest gene actually for risk of childhood asthma is a gene called ORMDL3, and that is an ORMDL sphingolipid biosynthesis regulator 3. And this was particularly important because there had also been work implicating sphingolipids in the generation of asthma. Now, this gene sits at the very beginning of the sphingolipid pathway. So the question is, can we understand something about how this gene or genetic variants in this gene that are already associated with asthma impacts the subsequent metabolism of sphingolipids that also have been known to influence asthma? Furthermore, in our study, since we have vitamin D, can we infer or see and understand anything about how vitamin D may impact this? So looking at this in a sequential way, one of the first questions we asked is, can we look at the ORMDL3 risk genotype in association with sphingolipids? And in this case, this just exemplifies what we see over and over again, is that increasing numbers of the gene, of the allele, are associated with a decrease, in this case, in the sphingomyelins. There are other specific metabolites in this pathway, and you'll see that are associated with a slow increase. And that is actually due to the metabolic flux of the pathway. The other thing we saw very distinctly, and note in these results, we did this not only in our cohort, but also in our COPSAC Friends cohort, who were able to replicate various aspects of this study, was this very pronounced interaction between this genetic variant and vitamin D. And that is with increasing numbers of the risk allele, when we see the effect of vitamin D on the genotype, we see that increased vitamin D and in combination with the non-risk alleles reduces the overall risk of asthma. So this then necessarily followed up with, can we actually understand the mechanism? And in looking at this further, what we were able to see is ORMDL3, as I said, sits at the beginning of this pathway and is responsible for potentially shutting off the whole pathway. what we see and what we're able to understand is if you don't have 2 copies of the risk variant, vitamin D supplementation will work and go through this pathway, increase sphingolipids, the good ones, and decrease asthma. However, if you had 2 copies of the risk allele, it didn't matter how much vitamin D you were given. This pathway, the ORMDL3 variant, would still shut off this pathway and really rendered any effect from vitamin D useless. We were also able to replicate this using human airway epithelial cells, where in this case, what we see is when we had increased amount of vitamin D, without the risk variant, we see that sphingolipid production goes up, which is what's responsible for the reduction in risk of asthma. However, in the overexpression of the bad allele, what we see is as vitamin D levels increase, we see a much more tempered relationship in the sphingolipid production, thereby further validating our result with more functional data. So in summary of this part, I want you to understand that we can utilize metabolomics in conjunction with these other omics and understand that sitting right before phenotype, at that critical stage, by linking the prior omics with this, it really enables us to incorporate outside influences like the environment in conjunction with prior omics to understand disease pathogenesis. And in all cases, we try to do this in a robust way where we are replicating results across multiple cohorts. So again, in this case, we see that metabolomics really can be a key identifier. These longitudinal measurements, identifying exposures, and replication are all key. So the next part of the study is going to take a shift and really ask this question: what can we utilize and understand about metabolomics and prevalent asthma in the context of potential precision medicine? And this study was done by a lot of people. Here's a bunch of members of my research team, where we basically utilized multiple studies in a discovery phase and then further studies in a translational precision medicine phase to study these data. The first step was taking 2 cohorts, one very large population-based cohort with almost 11,000 people, and look at associations with prevalent asthma. We then replicated that in a cohort that we actually generated within the Partners Healthcare Biobank. So we had a lot of comprehensive longitudinal data on these individuals, on their intake of different medicines over time, et cetera. So what did we see with that initial association with prevalent asthma? Well, this is just a negative log P plot of this, and what we actually see is this super high tail of highly significant results. And these results were significant in the discovery, and then also we saw some of these results in partners as well. But what could we actually see that was overlapping between the two, and how robust were those associations? So here you actually see the odds ratio in the discovery cohort and the odds ratio in the Partners Biobank. In the discovery cohort, we certainly see that these are tremendously robust associations with highly, highly significant p-values. These are all meeting Bonferroni adjustment. In the biobank, we also see significant associations, and these all were the FDR-corrected associations, so showing replication in both. But I want to talk about this. When I looked at these data, there's a problem I noticed that really was striking. And that is when we looked at the direction of effect of these different steroids, it actually went contrary to what we thought. And that is, if you look at all these odds ratios, the odds ratios for all of these steroid metabolites were associated with a reduction in risk of asthma. Now, this struck me particularly because, if anything, in this study, you would think individuals who are asthmatic are taking steroids. So if anything, we'd expect to see an increase in these steroids due to taking oral steroids or inhaled steroids. So why was it that we were seeing this very consistent reduction in steroids in all these individuals? So we asked the question, is this a potential effect of oral steroids? And is this a potential effect of inhaled steroids? Now, looking across here, we see these are all different steroid metabolites, and this is stratified by controls, asthmatics off steroids, and asthmatics on steroids. Now, these are the inhaled steroids. We found really zero association with oral steroids, but when we looked at inhaled steroids, as you see here, those on inhaled steroids had these dramatic reductions in all the various steroid levels. So this begged the question, what could potentially be happening here? And one of the questions is, with inhaled steroid use, are we actually getting some level of subclinical adrenal suppression? And what do I mean by that? Often with inhaled steroids, the research to date has really said that for most often, the effect is primarily in the lung. But are there more systemic effects than we realize? That is, are these going into the system and being absorbed and causing the adrenals to reduce their production of many steroids? And the primary steroid that really encapsulates a lot of adrenal function is cortisol. Now, one issue with this is, if we think of it, inhaled steroids really didn't become common until the late 1980s or 1990s. So in many ways, the long-term effects of these inhaled steroids haven't been studied. So what would we need to do to study this? And this really got me thinking about this idea of steps towards clinical translation. How do we go from a discovery all the way through to a clinical recommendation? And that is, we look at a cohort, we find something, we validate it, we identify the cause, then typically good science looks at this in the context of a randomized controlled trial. We quantify the clinical effects, evaluate the clinical impact, and then give a clinical recommendation. So thinking of this in that framework, we've already done the first two steps. The next question is, could we actually interrogate this in the context of a clinical trial? Fortunately for us, we had probably the best clinical trial in-house to look at this. And this was a clinical trial that randomized asthmatics to inhaled steroids and followed them for 4 years after randomization. Fortunate for us, we actually did have measures of cortisol and cortisone in this cohort. When we looked at these, one of the issues with this cohort is it was over the pubertal period. So we had to account for that. But sure enough, accounting for that, we see this dramatic reduction in both of these steroid levels throughout the course of the trial. Now, one of the issues here is that while all these data were generated using metabolomics, metabolomic data are not quantitative. That is, they are generated in a framework that the results are relative in any given study in which they're generated. So in order to really quantitate these in a robust way, we would have to utilize a better source for quantitation. And this brought us to this next step, which is this idea of, can we quantify this effect with clinical standards? And fortunately for us, we actually asked this question of, well, what is the clinical standard for looking at adrenal suppression? And in fact, the clinical standard for that is measuring cortisol in the context of an ACTH stim test. And we actually, in thinking about it, had these data in our biobank and electronic medical record system. So that is, we utilize the Partners Healthcare Biobank data and the electronic medical record data that has over 100,000 individuals, and asked these questions. We asked, can we identify individuals who have taken that or had that cortisol stim test. And can we look at that by asthma status and by inhaled steroid use? So this requires collecting a lot of data, and it requires tremendously querying the electronic medical record, cleaning the different data, generating phenotypic algorithms, harmonizing the electronic medical record data. So this is certainly quite a complex process. And in the case of what we did here, we were able to then go from the qualitative measures we had before to quantitative measures. We were able to query the EMR for adrenal suppression tests with the cortisol stim test. We were able to harmonize those cortisol levels in about 2,200 people. Now, this is important because when we think about a lot of these tests, something like the cortisol stim test, it is often incredibly difficult to get large numbers of people who've had this at one time because it's not that common of a test. But when we're able to utilize electronic medical records and biobanks, it really enables us to do this in a large scope way. When we have over 100,000 individuals, 2% results in 2,000 tests, which is what we had here. Again, we were then stratified by asthma status and inhaled steroid. And we asked the question, can we observe clinically important differences between those not on inhaled steroids and those on inhaled steroids, and those particularly who were asthmatics on inhaled steroids compared to other groups? Now, one other thing that is important to understand is cortisol by nature is something that has a diurnal variation. That is, at different times of day, cortisol levels peak and reduce. So when looking at cortisol levels, it is incredibly important to take this into account. The good thing about what we had in the biobank is because they recognize this, every cortisol measure that is ever done, the timing is very precisely calculated. So what we were able to do is to look at these cortisol levels for controls with no ICS, controls with ICS, asthmatics with no ICS, and asthmatics on ICS. And what we see here is that the most profound reduction, stronger than any others, is this joint combination of asthmatics on ICS. Looking into this further, what we do see is that asthmatics as a group already have a reduction in these steroids, which is one of the things that makes them susceptible to being asthmatic. When we then put inhaled steroids on top of that, it makes the suppression even worse because there's already issues with steroid production in these individuals as a whole. And you see that really reflected here. The other thing that was really notable is during the time frame, and this is 4 a.m. Throughout the day to 4 a.m., throughout this time period, the lowest measure or the lowest levels of these cortisol measures are actually during the time period when individuals are most susceptible to asthma attacks, making this even more important and imperative to address. So is this a clinically important drop? Yes, absolutely. So in looking at these steps towards clinical translation, can we evaluate the clinical impact? And the answer to that is yes. We also went further on and were able to understand through back calculating using the EMR that it turns out of asthmatics who are on inhaled steroids, and it really doesn't even matter how robust you are in that assessment, whether you say, oh, they've just been on inhaled steroids a shorter amount of time, or you're much more conservative and say, oh no, they had to be on steroids for 5 to 10 years. That pretty much a third of individuals who've been on steroids for really any period of length of time, who are also asthmatic, around 6 months or more, a third of those individuals go on to have clinically diagnosed amounts of adrenal suppression, making this certainly something that is relevant and important to be watching in more detail. So are we able to make a clinical recommendation? And I think that clinical recommendation is certainly And that clinical recommendation would be monitoring steroid levels if you're putting them on them, reducing the dosage or quantity as much as we can, and looking for signs of adrenal suppression before it gets to a point where it's irreversible. And another thing I want to point out when we look at these different groups, when we see this adrenal suppression, the other aspect that is very difficult about that is actually when the adrenal suppression becomes significant, it also makes these individuals at an increased risk of asthma exacerbations, highlighting another important point of why we would need to make this clinical recommendation. So in this case, I feel like we've gone through all of the steps from discovery to clinical recommendation. And to me, really, this is what I would identify as the steps and the translational omics standard that we need to be doing in all of our omics research. I want to end with just reflecting on a few things. I showed you this picture earlier of me in 1984. 1987 was a game changer for myself. That was when I was put on inhaled steroids. And since 1987, I have been on inhaled steroids literally almost every day of my life. So while talking about the dangers of inhaled steroids, I want us to also recognize that there's impact and that this has had a dramatic improvement on overall health of asthmatics and changing lifestyle. However, I want us to think of this in two ways. While I most certainly have some level of clinical adrenal suppression that is probably irreversible, I've now actually personally myself reduced the amount I'm on as much as I can. But this actually is a picture of my son who actually turns 10 on Monday. And what you see here is he unfortunately has followed in my footsteps, and very early on you see he had severe eczema and to this day has significant amounts of asthma. Now, what stopped this incredibly bad eczema he had as a kid? And that was steroid cream. And what does help his lifestyle now, and one of the things is inhaled steroids that he does need to be on. But the question I have for you is, while my generation, we may not be able to reverse some type of damage that could have occurred, can we think of alternate strategies in treatment where we're reducing these inhaled steroids so that lifestyle and quality of life is optimized while minimizing the impact for long-term adrenal suppression? So just to summarize, I want us to just think about when we think of multi-omics, just the potential that metabolomics has to bringing us at the forefront of omics in translational and precision medicine research. In this presentation, I've tried to show you how we can capitalize on things like study design, longitudinal data, generating quality metabolomic data, looking at multiple independent cohorts to maximize inference, linking this to biobanks and electronic medical records. Applying different novel approaches, or maybe approaches where we're just looking very focused, like we did in the sphingolipids, on one aspect of the multi-omics. And of course, working together through initiatives and consortiums. I'd like to end just by saying, showing this slide, one of my 4 children is a rock climber, and we watched this movie Free Solo, which is about a rock climber climbing up El Capitan with no support. One bad movement and he would fall and die. I suggest to you today that in research, we need to take the exact opposite approach. And it's the combination of collaborating together, good statisticians, doctors, biochemists, postdocs, collaborations, and the next generation that is really going to make us successful. So I would just say in the context of successful research and multi-omic research, there is no such thing as no free solo. So with that, I'd just like to thank all the people who were involved in this research and the various sources of funding. Thank you.